So it's a great pleasure to be here. I want to thank Tara for inviting me to speak to you today and for bringing me back to a place where I spent many, many hours listening to lectures as an undergraduate biochemistry major at Harvard. Being here again is a bit surreal and wonderful at the same time. My lectures in Biology 2, Hard to Believe, which I took in 1960 in this building. That course was taught by Jim Watson 2 years before he won the Nobel Prize in Medicine and Physiology with Francis Crick and Hugh Wilkins for You Know What. I also want to thank Tara for giving me the honor to speak to high school teachers. Our center is involved more and more with secondary school education, and I'd be glad to talk more about that. And also, my mother was a high school teacher, so I'm very happy to be here. So this being political season, the morning after, I want to open my talk with a story. It seems that Glenn Beck was invited by Sarah Palin to go bear hunting in Alaska when he was visiting her. And after he arrived, they got in her Hummer and they drove to a nearby mountain and hiked into the forest and waited with their rifles loaded. And after a while, a large brown bear came into view, and both Beck and Palin raised their rifles and aimed and fired. But at that time, both triggers of the rifles jammed at the same time. The bear saw them and started charging toward them. And in a panic, they began to pray in unison. And they said, "Dear Lord, we have always been your faithful servants. Dear Lord, please, please make that bear a Christian." And just at that moment, the bear came to a screeching halt, and it put its huge paws together and looked at the sky and said, "Dear Lord, I thank thee for the gift I am about to receive." So, as you can probably tell, I'm sometimes rooting for the bears, even though two black bear completely demolished my beehives 3 years ago. But I'm rooting for them because they're truly remarkable creatures, very valuable to human medicine, as you will soon hear. So, as Tara told you, in the 1980s, with 3 other Harvard faculty members, we started this organization, the International Physicians for the Prevention of Nuclear War. The most important contribution by the tens of thousands of physicians around the world who were eventually part of this federation was to help people grasp what a nuclear war would really be like, so that they knew that these weapons were so destructive that they couldn't be really used in wartime, and so that they would do everything in their power to prevent a nuclear war from occurring. And we did this by translating the abstract and technical science of nuclear weapons explosions, the physics and chemistry, etc., into concrete, personal, everyday language of human health that people could relate to and understand. Namely, what would really happen to people if there were such a war? What would happen to hospitals? What would happen in terms of blast injuries and radiation sickness and burns? What would happen to the food supply and the water supply? What would happen to the blood supply? Could you even get to hospitals? And by talking about nuclear war in these very concrete, graphic terms, I think we in some ways help change public opinion and maybe even public policy. But in contrast to the science of nuclear weapons explosions, with changes to the global environment like climate change or loss of biological diversity, the level of complexity and abstraction are orders of magnitude greater than they are than talking about nuclear weapons explosions. These changes occur very slowly and on global scales. They're often very hard to see over normal fluctuations in things like temperature or precipitation patterns, or what species are present in a certain environment and which ones are not. These are very specialized and hard to see phenomena, unlike a nuclear weapon explosion. And it's particularly hard for people to see, those of us who live in wealthy industrialized countries or those of us who live in cities. How can one walk around Cambridge, for example, on a beautiful summer day with all the flowers in bloom, birds overhead, trees leafed out, and think that anything is wrong at all with the world, with the environment. And it is therefore, it seems to me, even more essential for physicians and public health professionals to be involved in helping the public understand the implications of these changes in very concrete, personal, everyday language for our lives. We have no Hiroshimas or Nagasaki's to serve as models, and the number of variables that are involved compared to a nuclear weapons explosion is so much greater. And the task is made that much more difficult because there's such a fundamental misunderstanding, when you think about it, that most people have about the environment, that human beings are separate from the environment. The environment exists outside of us. And so as a result, I think people feel that we can damage or degrade the oceans or the soils or the atmosphere, and even lose countless species in the process, some of which have been on this planet far longer, for hundreds of millions of years longer than we have, like amphibians or sharks or horseshoe crabs, as if these changes would have no effect on us whatsoever, almost as if they were happening somewhere other than where we lived. And this, in my view, is really at the heart of the global environmental crisis. So because I'm here with teachers, I want to say a few words about education in a more general way before getting on to the topic of my talk. I think this point of separateness from the environment is one of the most important things that we need to deal with in educating children. And these lessons should be given, in my view, as early in a child's education as possible. They're as important as reading and writing and thinking abstractly and doing math and science, to provide them with the knowledge that human beings are an intimate, interdependent part of the living world, and that we cannot damage this world and the other organisms we share it with without severely damaging ourselves. And so I want to talk more about that in a bit later. I have some other thoughts about education. I just want to throw them out since I have the opportunity and a captive audience. I know there's not time to discuss them, but some other things I've been thinking about. You heard Tara mention that I'm a psychiatrist. I think one thing that we need to talk about is how children, how human beings are a part of at least three different human communities. The one in which we live, obviously, our neighborhood, our town, our state, our country, but also being a part of the world community. And finally, being a part of the community of people who came before us and those who haven't been born yet, all of those who will live after us, and that we have a responsibility as human beings to care for all of these communities, particularly, obviously, for the disadvantaged, to provide for them good health and the same opportunities we have had, and the biologically rich world we have been privileged and blessed to live in. Two more final comments about education. In these days where young people, and many who are not so young, can't seem to be conscious and not be connected to their iPods or cell phones or email or blogs or Facebooks or Twitter accounts, I can't think of anything more important than to provide our students with the experience of quiet time, with reflection, with being alone with one's own thoughts, with being happy and being able to enjoy one's own company. As a psychiatrist, I worry a lot about the implications of people not being able to be alone, particularly in terms of their developing intimate relationships. They have to be with someone else or connected to someone else rather than they want to be. And finally, just one other quick point. In these days of information overload on the internet, when the careful and considered conclusions of experts are buried in an avalanche of wrong information, some of it quite deliberately wrong, we need to teach our young people how to distinguish between real expert opinion and all the junk that is out there. And the internet is a totally wonderful thing. I can't do without it. But I believe in some ways it's the reason why people are confused about things like climate change or vaccinations or all kinds of other things, because there's so much other information they're getting. Now, on to the topic of my talk. My parents told me many things about being a person of good character, but two that have always remained with me were, one, never ask other people for money, and two, never be guilty of blowing your own horn. So clearly, I have not listened to them. I do the first every day as director of a nonprofit, the Center for Health and the Global Environment. In this economy, I do it more than once a day. And as you shall soon see, I will be doing the second this afternoon. So you must please forgive my blatant and shameless act of self-promotion as I tell you about our new Oxford University Press book, Sustaining Life. But I and the more than 100 other scientists who help from all over the world who helped put this book together are enormously proud of it. And I can honestly say I have never worked harder on anything in my entire life. So before starting, I want to recognize my fellow editor and lead author, co-author, Aaron Bernstein. There wouldn't be a book without this brilliant young physician. So this is Art Wolf's spectacular photo of what's called a blue dart poison frog. Its Latin name is Dendrobates tinctorius. It lives in Suriname and other parts of South America. We chose this photo not only because amphibians are among the most endangered group of organisms on the planet. Some 1/3 of the known 6,600 species are at risk of extinction, but because they're also enormously valuable to human medicine. The alkaloid toxins in the skin of this frog, for example, are very useful for understanding how local anesthetics like Novocaine that we get at the dentist work. But we also chose this picture because the frog seems to be admiring our title, which we liked. And if you look very carefully, it's probably hard to see, you may even notice that the frog is smiling because it notices that the world's leading ant biologist, Edward O. Wilson, who studies some of Tinctorius's favorite food, has written the foreword to this book. So, title page of the book, we wanted to involve the United Nations, and we were very pleased that 3 of the agencies, UNDP, United Nations Environment Program, and the Convention on Biological Diversity all co-sponsored the book, and that Kofi Annan wrote the prologue. So I see many of you have cone snail shells in your bags. This is the front plate of the chapter on medicines from nature, chapter 4. It was a real challenge to decide how to organize this enormously complex and comprehensive topic. Of course, we wanted to cover what has been most discussed and written about, namely when you burn down or cut down tropical rainforests, you may lose medicines from higher plants in those rainforests. But we also wanted to make sure we covered plants outside the tropics, in temperate zones, temperate forests, and plants not in forests at all, and animals in the tropics and in other parts of the world, and microbes, both on land and in the ocean. So this chapter tries to take a look at the wide range of medicines that have been discovered from various organisms on our planet. We had to talk about biodiversity and medical research. You all know this guy. This is a fruit fly. This is a scanning electron micrograph that's been colorized, Drosophila melanogaster. This chapter looks at some of the contributions that various organisms have made to medical research, to our knowledge about how our bodies work in health and disease. You know that Drosophila has provided some of the very basic genetics information that has allowed us to really sequence the genomes of organisms, including the human genome. Without that knowledge, we wouldn't be able to do that without Drosophila, those early insights that we got from that. Now, the laboratory organisms in this chapter are not endangered by any means, but we're talking about them to illustrate the kinds of totally unique information, physiologically, chemically, anatomically, that organisms can provide us to give us a window into understanding ourselves, making the point that organisms in the wild, most of which, by the way, have not yet been identified, may contain similar insights and valuable information to help us understand ourselves. Can't talk about biodiversity and health without talking about infectious diseases. I'll talk more about that in a moment. These are hand-drawn images of Anopheles mosquitoes, 4 different species. This is Anopheles gambiae, which is the principal vector of malaria in Africa. This chapter looks at how changing biodiversity of vectors of infectious disease, like these mosquitoes or ticks, of the pathogens that they carry, and of the hosts that these pathogens live in before they're transmitted to us, how changing those relationships affect the emergence and spread of infectious disease. So that's the chapter on that. Can't talk about biodiversity and health without talking about food. This is the front plate of the chapter on biodiversity and food production. This is a cross-section, idealized cross-section of temperate soil, just illustrating the enormous breadth of life that lives under our feet, both at a macroscopic level, like moles and earthworms, but at a microscopic level, mites and nematodes, and at a really microscopic level, protozoan and fungi and algae and bacteria, et cetera. Making the point that soil fertility is very much dependent on the interrelationships of these organisms, by the way, which is very poorly understood. We know more about perhaps the surface of the moon than we know about what's under our own feet, particularly at a microbial level. Can't talk about biodiversity without talking about ecosystem services. This is a very moving picture for me. These are women at what's called the Maotian County. It's on the border of China and Nepal. They are hand pollinating apple blossoms with these little sticks that have brushes at the tip. It takes 25 people to hand pollinate 100 apple trees, a job that is done with much greater efficiency and effectiveness by 2 beehives. And the bees in this area have gone extinct because of an overuse of pesticides. So that's why they are doing this. Again, making the point that ecosystem services we take for granted. We don't even think about them, but they provide everything that supports life on this planet. And this chapter looks at those services. So that's 5 of the chapters in the book: biodiversity in medicines, biodiversity in medical research, biodiversity in infectious diseases, biodiversity and ecosystem services, and biodiversity in food. But we wanted the book to be provide a really complete introduction of this subject. We wanted it to be used as a textbook. We wanted it to be a reference for scholars in medicine and ecology. And we wanted it also to be a resource for people interested in the environment and in human health. So as a resource for experts, it needed to review the literature. It's part of what took so long. We had to look at 1,400 different references in this field, and the bibliography, I think, contains the largest collection of those references that has yet been published, some 70 pages of them. We wanted it to be used as a textbook, and therefore we had to add sort of basic information about biodiversity, which I'll talk about in a moment. And we wanted it to be a resource for environmentalists, for policymakers, many of whom would not have a technical or scientific background. So it had to be written in everyday language. No easy task for scientists, as you can well imagine. So many schools, including Harvard, are using this book as a textbook in some core courses. This is the main textbook for what's called OEB 10, Organismic and Evolutionary Biology 10, Foundations of Biodiversity, which biology majors need to take. It's also a text in core courses at University of Wisconsin, Wellesley, Brandeis, Cornell Vet School, and some high schools, including the Buxton School and the Belchertown High School and others. So let me go through some of the other chapters. We wanted, as I mentioned, the book to be comprehensive. So chapter 1 is what is biodiversity? You can't really ask that question without showing an image of beetles because there's some 350,000 known beetle species, some 6 times the number of all vertebrate species combined. And when the great famous British evolutionary biologist J.B.S. Haldane was asked what he could deduce about the creator from having studied his creation for his entire life, he was purported to have said, quote, "He had an inordinate fondness for beetles." Chapter 2, how is biodiversity threatened by human activity? This may be a little hard to see. It's a satellite photo of the Gulf of Mexico. This is the isthmus of the Mississippi River, delta emptying into the Gulf. These are cloud formations. And the reason that this is the front plate of that chapter, how is biodiversity threatened, is that we wanted to illustrate sometimes the complexity, the enormous cascade of events that can affect biodiversity. In this case, the Mississippi River is emptying and carrying down the effluent from discharge of nutrients from farms all along the Mississippi, from sewage discharge by cities into the Mississippi, particularly true when there are extremely heavy rain events, which may have occurred in 1993 when, you know, the Mississippi River flooded its banks. But it carries those nutrients down, empties them into the Gulf along with sediment, which is the brownish area. But also those nutrients then lead to algal blooms. And from what you know about algal blooms, when the algae die, they deplete oxygen in the water column, and these become dead zones. So after these very heavy rain events, the dead zones in the Gulf enlarged, and this is now an illustration of how an enormous range of events can affect biodiversity. So this chapter looks at pollution, of course. It looks at climate change. It looks at invasive species. It looks at all the general factors that lead to a loss of biodiversity. The next chapter is really the heart of the book. This chapter examines 7 groups of organisms that in fact are threatened, are endangered, and reviews the contributions that they have already made to human medicine and those they may be expected to make if we don't wipe them out in the meantime. So I'll get back to some of that in a bit. You can't talk about biodiversity in food without talking about the very controversial and complicated subjects of genetically modified organisms and organic farming. This is my friend Fred Kirshenman standing before his 3,700-acre organic farm in North Dakota. This is buckwheat in the foreground in bloom. These are his hedgerows in between his fields. Fred has been doing this for decades. What's interesting is that his yields in good years are almost the same as his conventional farming neighbors growing the same crops, but his yields in drought years are far greater. Those of you who do any gardening or farming, you know that when you build up the organic matter in the soil, it holds on to water much better. And that has been Fred's discovery, not his discovery, but his experience. And organic farming is, I think, going to become increasingly important as energy becomes more and more expensive and as we're more and more concerned about climate change. It's much more energy efficient, uses much less petroleum products, which, as you know, are made into fertilizers and pesticides, et cetera. The last chapter is how individuals can help preserve biodiversity. This is a man in Haikou, China, recycling plastic bottles. This chapter looks at all the various things that people can do in terms of purchasing food and saving energy, et cetera. So what I thought I would do with my remaining time is go through some of the case studies in the book to illustrate some of the points that I have just summarized. So the first is about polar bears. So these magnificent creatures, they're the largest land carnivores. An adult male standing on its tiptoes can be 11 feet tall, weigh 1,300 pounds. They evolved roughly the same time that we did. Around 190,000 years ago, they evolved from brown bears. That's the same time our species evolved, roughly 190 to 200,000 years ago. It's predicted, as you know from following the news, that they will be extinct in the wild by the end of this century, if not before, in part because they're at the top of the marine food chain. So their main food is seals, and seals are at the top of the marine food chain just below polar bears that eat large fish, that eat smaller fish, that eat smaller fish. And as you know, going up the marine food chain, pollutants like PCBs and other long-lived organic pollutants are concentrated, and polar bears then get a very high dose of some of these compounds which affect various organ systems. But the main threat to polar bears is global warming and melting of the sea ice in the Arctic. So what's happened is, and you can follow this with satellite photos, this is very well documented. Polar bears' main food is seals. They have extremely good hearing. They're very good swimmers. But they mostly get seals by wading at thin areas of this ice for seals to come up for air. They're marine mammals, they breathe air, they have to come up. And polar bears wait at these thin areas of ice, and when the seals emerge, they grab them and eat them. So as you can imagine, if there's open water all over, the seals can come up for air and elude capture by polar bears. And this is now being very clearly documented. Polar bears are starving or not getting the nutrition they need. They're not having the number of young that they usually produce, and their numbers are dropping. So let me go back. So there's been a lot of discussion and a lot of anguish about losing polar bears. The baby polar bears are the cutest things in the world, so environmental groups have shown them widely. And, you know, we're heartbroken because this has become an iconic image of what we will lose with global warming. But the medical value of polar bears is almost never mentioned. Let me tell you a bit about that, because it's fascinating stuff. So like all bears that hibernate, and they actually don't truly hibernate. It's called denning instead of hibernating. So true hibernators are ground squirrels and woodchucks and bats. So they enter into a very depressed metabolic state. Their heart rate drops significantly, their body temperature drops. They enter into a deep torpor. So denning bears also, heart rate drops, temperature drops, their metabolism drops. They enter into a kind of a torporous state. But as some bear biologists who study them in their dens have discovered it to their dismay, they're fully arousable and very quickly. So like all bears that hibernate, this is a black bear, and you can see its eye is glazed because the biologist has gone into their den and fired an anesthetic dart and put the mom to sleep. Bears also often give birth in the den. So like all denning bears, polar bears are largely immobile for several months at a time, 5 to 7, sometimes even longer months. Black bears too, and other hibernating bears. And yet they don't get osteoporosis. Every other mammal, including us, during prolonged immobility starts losing bone mass. So all the time, if we were immobile for a period of 5 months or longer, we would lose 1/3 of our bone mass. So all the time there's a dynamic process that goes on in all of us. There's cells called osteoblasts that are making new bone. There's cells called osteoclasts that are resorbing bone. So the bone architecture is constantly being remodeled. It's not a static thing. It's a very dynamic process. So under conditions where there's no muscle pull on bone, so immobility, people who are paralyzed, people who are bedridden, inactive elderly, particularly postmenopausal women, because estrogen has a positive effect on bone formation, we tend to lose bone. This is an enormous public health problem. It kills 70,000 people in this country every year, costs the US economy $18 billion. For those of you who have osteopenia or osteoporosis, you know that you don't reverse the bone loss. You can stop it in its tracks. I have mild osteopenia. I take a medication called Fosamax, which is what's called a bisphosphonate. So it stops bone loss, but it doesn't put back new bone. So this is a largely incurable disease. Denning bears don't get osteoporosis. They have circulating compounds in their bloodstream that inhibits osteoclastic activity. They absorb their calcium. They actually lay down bone during prolonged immobility despite not eating. They also don't urinate for months and months at a time. If we don't urinate for a few days, we're dead. We have to get rid of our urinary waste. They're toxic to our system. People with renal failure, there is no cure for renal failure. We can only remove those compounds by renal dialysis or by a kidney transplant. There's no medicine you can take when your kidneys aren't working. They don't get toxic despite not getting rid of their urinary waste. They resorb them in their bladder. They break them down into ammonia. They convert them to amino acids, and they make new protein. They also don't lose muscle mass, even though they're not moving. So they are physiologic phenomena. Nobody understands how they do that. Polar bears, in addition, become massively obese prior to entering their dens because they have to survive on that fat for those months at a time. And yet they don't develop type 2 diabetes, which we do when we become massively obese. Again, nobody understands that. We need to study them in the wild. Bears don't hibernate or don't den in zoos. We need them in the wild. So if we lose them, we will lose some of these secrets that may hold the insights for treating 3 largely untreatable and very costly human diseases. 250,000 Americans die every year from obesity-related type 2 diabetes. It's virtually epidemic. More than 20 million people in this country have that condition, 7% of the population. And more than 85,000 people die of renal failure. Costs the US economy $35 billion a year. So let me go on to another topic, a plant, Pacific yew tree. Because I mentioned before, you recall, that it's not just plants in the tropics, but also plants in temperate zones, which can lead to very valuable medicines. The Pacific yew tree, I'm sorry I don't have a picture of the tree, but you all know about yews, these little red pink berries that we all threw at each other when we were kids. This tree lives in old growth forests of the Pacific Northwest. It was routinely burned and discarded because it's a runted, kind of misshapen tree. It lives amidst Douglas fir trees, which were being logged for their beautiful straight wood, gorgeous trees, until the United States Department of Agriculture and the National Cancer Institute did a plant screening looking for medicines, particularly cancer medicines. And they found in the bark of this tree a very unique compound, very complicated ring structure, which chemists in the lab could not have just synthesized, very complex molecule. Has 2 to the 11th possible structures, for example, this one ring. And in lab tissue culture tests, they found that it had very potent cancer chemotherapeutic activity, particularly against ovarian cancer. In fact, it was the most effective agent for inducing remission in cases of ovarian cancer. And it did it by a new mechanism that no one knew about before. You all recognize this slide of chromosomes lining up on the metaphase plate, and they move, as you know, to the mitotic along the spindle to the poles, and then the cells divide. Other cancer chemotherapeutic agents act often at the formation of the spindle. This particular drug bound to the tubulin, the protein of the spindle, and prevented the disassembly of the spindle, and therefore stopped cell division. It led to a whole new class of cancer chemotherapeutic agents called taxoids, which are among the most effective agents in treating cancer of the ovary, as I mentioned before, breast, prostate, and lung. Enormously important drugs that came from this runted tree that was routinely burned, not in the tropics, in our country, in Washington state. Taxoids also have another very important use. So this is an arterial stent. You may know that when the opening of an artery is blocked by arteriosclerotic plaque, they insert a catheter and then blow up the balloon in the catheter, expanding this metal basket, and then they remove the catheter and the balloon, leaving the metal basket in place to open up the arterial opening so that blood can pass. But the problem has been that whenever you have a foreign body, the body treats it as an invasive agent and tries to heal it, tries to get rid of it. And what happened is the cells lining the arterial wall, endothelial cells, started multiplying and growing over and inside the stent, blocking it so blood would not flow. So they started, a light bulb went off, they started coating these metal baskets with drugs like Taxol to prevent the endothelial cells from dividing and keeping the stent open. Enormously important medical use, again, coming from this runted, the bark, a chemical in the bark of this runted tree. Just two more examples, moving to the oceans. You know about coral bleaching. Corals live at a very narrow temperature range, and when that is exceeded even by a few degrees for a few days, they undergo what's called bleaching. This is a coral reef off the Florida Keys in June of 1996 and October of 97, the same reef showing bleaching. So corals live in symbiosis with algae, or actually zooxanthellae. They photosynthesize, and that gives them their color, but they're actually sort of part animal, part plant. And when the temperature warms, the zooxanthellae leave the corals and they look bleached. This coral is still alive, but in a prolonged bleaching episode, they can die. But what's often happens is that it affects their immune system because the zooxanthellae provide sugars and oxygen to the coral, which they need, and they become weakened and they succumb to infection by bacteria and fungi, etc. So coral reefs are enormously biodiverse. They're called the rainforests of the sea. And I'm going to go back, as I mentioned, to talk about these organisms that you have in your bags, cone snails. So this is a cone snail called Conus bullatus. It's sampling the water with its siphon to detect organisms because they are predatory snails. They're a very large group of predatory snails, and they defend themselves and they eat by firing a poison-coated harpoon at their prey. This is a blow-up of what those harpoons look like. What's amazing is that each species makes a slightly different harpoon. Any of you have seen whale harpoons or eel harpoons. You notice that the design is essentially the same. They go in, but you can't pull them back out. So that's the shape. And so they paralyze their prey, and then they bring them into their enlarged stomach and digest them. So it's an evolutionary explosion. They're only about 50 million years old. There's some 700 different cone snail species. But even more remarkable is that each species is thought to make 100 to 200 different and distinct poisons, toxins. They're peptides, small proteins like the peptides in scorpions and spiders and sea anemones. But cone snail peptide toxins are much, much more numerous. There may be 140,000 of them, and they're much more potent and they're much more selective. They seem to work by binding to cell membrane receptor sites on all animals, including almost everyone we know about in ourselves. There are only 100 that have been studied in any detail, and only 6 species have been studied out of 700 in any detail. And already, these toxins that bind to acetylcholine, serotonin, norepinephrine, vasopressin, calcium channels, sodium channels, etc. And all the subtypes that we know about are being studied by pharmaceutical companies because, as you know, it's the binding to a cell membrane receptor site that is affecting the activity of those cells and therefore the organisms in which they find themselves, whether it's the heart or the nervous system, pancreas, etc. So many of these drugs are in clinical trials, and one is already on the market. Let me tell you about that one. It's called Prialt. Prialt is a calcium channel blocker that comes from one of these peptides. It's been synthesized so that it's the exact replica of the natural substance. It is the most important painkiller that's come along in a very long time. Now, in medicine, the holy grail in pain relief is to find a drug that's as potent as the opiates, but which doesn't cause addiction, but that's not the big issue. It doesn't cause tolerance. So tolerance is the big problem in pain relief. Any of you have ever had morphine after you've broken a bone or had surgery. You know how effective it is. It's a remarkable drug. It's an amazing drug, but it works acutely. It's not such a great drug for people who have chronic severe pain, because what happens is you get tolerance. You adapt to, essentially, chemically, you adapt to the dosage, and you have to up the dose to get the same effect. And then you have to do it again if you're using morphine chronically or other opiates. And of course, addiction occurs, but it's the tolerance that's the problem, because eventually you reach other side effects like respiratory depression or complete paralysis of your bowel or stupor, a state, and so you can't go beyond that, or it just doesn't work very well anymore. So the holy grail is to find a drug that's effective but doesn't cause tolerance. And this drug, Prialt, that comes from a cone snail toxin, does that. It's 1,000 times more potent than morphine, but doesn't cause tolerance. An amazing discovery from this little snail that lives in coral reefs of the South Pacific. There are other drugs in clinical trials that are potassium channel blockers that seem to protect heart cells when there's not adequate circulation. There's another calcium channel blocker type that is being used that protect neurons in the brain when there's not enough circulation, like after a head injury or a stroke or open heart surgery. These are all in clinical trials. Cone snails are thought to perhaps contain more medicines for human diseases than any other group of organisms on the planet. Maybe 140,000 of them. And yet they live in coral reefs. Let me talk about an infectious disease and then start winding down. So this is a map of Lyme disease cases. Each of these dots is a Lyme disease case, and you can see the tremendous concentration in the middle Atlantic states, eastern Pennsylvania, New Jersey, parts of Maryland, and of course southern New England. You can see Long Island here. All of Massachusetts is black. You can even see Cape Cod and Martha's Vineyard in Nantucket. So a big concentration in the Northeast, a concentration in the upper Midwest, sort of smattering of cases in the Southeast, almost nothing in the West for a couple of reasons. One is this is a population effect. There are big open areas here. And then cases in California and the upper Pacific Northwest. So if you look carefully at these distributions, you'll notice an interesting thing, and that is that they pattern the blue and red states of the 2004 election. And this has caused some people to wonder whether maybe Lyme disease has a positive effect on the portion of the brain that controls intelligence and judgment. Others, of course, have included the opposite. This is completely bogus. I just wanted to see whether you were paying attention. You are. So Lyme disease is the most common vector-borne disease in the United States. Causes 20,000 known cases. There may be an equal number that are not registered. Any of you have had Lyme disease in this room, or know of people who've had Lyme disease? So you know that the early symptoms of Lyme are very subtle. You feel like you have a flu, you're kind of achy, you don't feel very well. You may have a rash, but only 75% of people have that characteristic bullseye rash. You may be able to discover the tick on you, but you know the ticks are very small, especially the nymphs, which are the ones that mostly carry the bacteria, and sometimes they don't cause a local skin reaction. They're very hard to see. The Lyme disease blood test is often negative at the beginning. It turns positive later on. So this is not an easy disease to diagnose. And what I do, I get embedded ticks a lot, because our farm in Central Mass is a prime Lyme area. I just take doxycycline as soon as I have an affected tick, because I don't want to risk getting Lyme. This is a serious illness. Not so much in the early stages, and it doesn't cause fatality, but it causes chronic disease of the heart, of the nervous system, and of the joints, which is harder and harder to treat when you wait longer. It's interesting, Lyme is caused by a bacteria called Borrelia burgdorferi, and it's a spirochete. And if you remember the other spirochete bacteria that causes disease that is often very hard to diagnose, it's syphilis. Often can masquerade as many different kinds of disease. It's a systemic disease. So one of the things that was noticed is that in areas where there's little vertebrate diversity, there's a high risk of getting Lyme disease. And no one could kind of figure out why that was. But some very elegant field studies determined why that may be so. And that is that the vector for Lyme disease in the East Coast is this organism, the eastern black-legged tick. And the eastern black-legged tick is an omnivorous feeder. It bites anything that crosses its path, our cats, our dogs, us, birds, mice, squirrels, chipmunks, even bites reptiles. So we are a host for Lyme disease. We get Lyme disease. It's a systemic disease. But what's interesting is that even though we get Lyme disease, we're in what's called an incompetent or even a dead-end host. And what that means is if another tick bites us when we have the disease, it can't pick up that infection and pass it on to another organism. So if you have a lot of vertebrate diversity, you have a lot of incompetent hosts like us, like our dogs, like other organisms, that don't keep the disease cycle going. By contrast, this is an extremely competent host, the white-footed mouse. And this host actually does very well in degraded environments, in fragmented forests, in field communities. It proliferates widely. It has very high reproductive rate. It survives quite easily on not much food, acorns and other food. So if you have a degraded environment, you decrease vertebrate diversity, but you don't decrease in this part of the world the population of the most competent host, where the Lyme bacteria proliferates in its bloodstream and another tick bites it, it passes that infection on to other organisms. So with degraded forest, with fragmented forest, you decrease vertebrate diversity and you do what's called provide a dilution effect. That is, the pathogen gets diluted in all of these incompetent and dead-end hosts like ourselves that doesn't keep the disease cycle going. Is that clear? So the other thing that happens is that if you have a lot of vertebrate diversity, you have a lot of organisms that compete with the most competent host for food, like other rodents that are maybe not as good hosts as the white-footed mouse, or deer, or other, forget about deer, that's more complicated, but other organisms that compete with it and keep its population down. And therefore, again, there's a lower risk of getting Lyme for us in those habitats. And finally, you have organisms that are predators of white-footed mice, weasels, foxes, bobcats, fishers, eagles, snakes. that eat these guys like Godiva chocolates. They're the most delicious thing you can imagine for a snake. So again, keeping the population of the competent host down and making it less likely that we will get Lyme in those environments. So here's a very elegant demonstration of increased biodiversity and a lower risk of a major human infectious disease. So I want to end by talking about three stories about amphibians. I started with amphibians. I'm going to end with amphibians. For those of you in the front row, you'll notice I'm wearing an amphibian bow tie. So I'm going to start with this one. This is the waxy monkey tree frog. It's hard to believe this is a living organism. This is not a rubber model of a frog. Its Latin name is Philomedusa savogi. It's found in dry prairie regions of Argentina and Brazil and Bolivia, Colombia, Paraguay. So frogs have antimicrobial compounds secreted by their skin that protect them from infection. And they're exposed to a wide variety of bacteria and fungi and viruses. And in this particular frog, there's a class of compounds called dermoseptins, which are very potent antimicrobials for some of the fungi that can kill people with HIV/AIDS. So there's a lot of interest in those particular compounds. But even more important than these dermoseptins, frogs and other organisms that have these innate immune systems, that is, not the kind of adaptive antibody kinds of immune systems that we have, they have made these compounds and defended themselves against infection for thousands and millions and even hundreds of millions of years. So think about that for a moment. These antibiotic strategies have worked without the development of antimicrobial resistance, without the development of antibiotic resistance. So that is a huge problem in medicine now. Those of you who have followed this, there are TB strains that are resistant to almost all the antibiotics that we have. There are gram-negative bacteria that are rampant, I'm sorry to say, in hospitals, even in intensive care units, types of bacteria called Klebsiella or Acinetobacter, which are effective against in preventing themselves from being attacked by antibiotics. The race is on to develop new antibiotics and new strategies for treating bacteria which can evolve resistance very quickly, and very big worry in medicine now. So we need to understand how these organisms have had effective means of preventing infection without the development of antibiotic resistance. There's some clues. Some involve the fact that their antibacterial attacks cell membranes rather than cell walls, or antibiotics like penicillin attack bacterial cell walls. It's much harder for a bacteria to evolve resistance by changing the structure or function of its cell membrane as opposed to its cell wall. They also release a cocktail of different compounds at once, so it's much harder, and they vary, much harder to develop resistance against many different ones. But we need to understand that much better, and frogs offer a very important, and toads, very important model. This organism is also a live organism. This is called the crucifix toad, or Notoden bennetti. And it's called a crucifix toad because the black markings on its back are in the shape of a cross. This organism is found in southeastern Australia. It lives for as long as 9 months of the year underground in dried mud. Doesn't eat, doesn't drink, lives there in this suspended state. It emerges only during very heavy rains, and when it emerges, it's very vulnerable to insect attack. Its only defense is to make this very proteinaceous, sticky glue on its back. So like roach motels, the insects stick to them and they can't get out. That's its only defense. So people got very interested in this glue because it is very strong and it works even when it's soaking wet. So the search in medicine, in surgery, for a biological glue has again been a very important search because you want to get a glue that sticks tissue together until the body can heal it itself. So we have biological glues like albumin, for example, but they're not very strong. They don't work with, say, a tear of the cruciate ligament in your knee, for example, like happened to poor Tom Brady, who seems to be doing pretty well again. And you could use superglue, that's strong enough, but superglue is toxic, it's also brittle. But most important, the reason to get a biological glue is you want a glue where fluids and gas and cells can move through the substance of the glue so that the natural healing mechanism of your connective tissue, your fibroblast, can heal the wound and make a very strong healing. So it turns out that this glue is strong enough. And with experimental tears of the ligaments in the knees of sheep, surgeons have found that this glue can hold those tears that are subject to huge torque energy together. So great interest in this bizarre little organism that lives in Australia. One last story to illustrate the kinds of information that we can get from other organisms. This is what's called the gastric brooding frog. 2 species of gastric brooding frogs were discovered in the rainforest of Australia. The female swallows the fertilized eggs, which then hatch in her stomach. There they develop into tadpoles, and when they reach a certain stage of development, the mother vomits them out, as she's doing here, into the outside world, into an aquatic environment, where they continue their development into adulthood, and they become sexually mature, and they mate, make eggs, and the whole cycle starts all over again. So all vertebrates, including amphibians, including us, begin the process of digestion in our stomachs. When food enters the stomach, there's a cascade of events where substances are released to release enzymes and acids to start breaking down food. And after a certain point, another cascade of events empties the stomach into the small intestine so that food can be continually digested and provide nutrients. So it was discovered, not surprisingly, that the eggs and the newly hatched tadpoles secreted a substance or substances that prevented their being digested, that inhibited this process of acid and enzymes that would break them down, not surprisingly. So there was a lot of interest in this, and you can imagine why. There was a lot of interest because these compounds may have worked by a completely different mechanism from the ones that we know about. So peptic ulcer disease in the United States, at some point in their lives, affects 25 million people. So we have various drugs to treat peptic ulcer disease or hyperacid stomach, proton pump inhibitors that stop the acid, Prilosec and others that people can take. They have many problems, but they're fairly effective. But these compounds may have worked by a completely different pathway and may have been much more effective. So the search was on to identify what they were, but the research had to stop. Both species of gastric brooding frogs, the north and southern gastric brooding frog in eastern Australia, the only ones known about anywhere in the world, went extinct. Partly probably because of damage to their stream and forest habitats, probably partly because of global warming, because the temperatures in those forests increased, and ultimately perhaps because they got these fungal infections called chytrid infections, but human events in all cases. And think about it, the compounds were never fully characterized. So we will never know what they were, and we will never know how they worked. Those compounds are gone forever. So this is our center's website. Everything we do essentially in our center is here. The course we taught at Harvard Med School for a decade, the course we taught for congressional staffers for another 10 years, lectures we give, publications, et cetera. Almost everything is there. And I'll be glad to, including the high school curriculum, I think some copies of which are outside. Tara, is that correct? But lesson plans and things like that are on the website. We're very interested in refining that. We're in the process of putting together a job description for a director of secondary school education. We still teach a course at Harvard School of Public Health for grad students, but we're very interested in bringing these materials to a younger and younger age group. And let me close by telling a really amazing story. It's very hard for you to see, but this is a ring of Saturn. This is another one. And you can barely see it, but there's a very small dot in the middle of this ring. So this image was taken by the Voyager 1 spacecraft on February 14th, 1990. The Earth was over 4 billion miles away. At the suggestion of Carl Sagan, NASA directed the Voyager to turn around and photograph the planets of the solar system. This is all online, by the way. You can find it. So one image showed what Sagan called the pale blue dot. And I want to read what Carl Sagan, whom I was very lucky enough to know, and who died tragically at a very young age, said about that pale blue dot, because I think it's one of the most profound things I have ever read. He said, quote, "Look at that dot. That's here. That's home. That's us. On it, everyone you love, everyone you know, everyone you ever heard of, every human being who ever was lived out their lives. The aggregate of our joy and suffering, thousands of confident religions, ideologies, and economic doctrines, every hunter and forager, every hero and coward, creator and destroyer of civilization, every king and peasant, young couple in love, mother and father, hopeful child, inventor and explorer, every teacher of morals, corrupt politician and superstar, every supreme leader, every saint and sinner in the history of our species lived there on a mote of dust suspended in a sunbeam. The Earth is a very small stage in a vast cosmic arena. Think of the rivers of blood spilled by all those generals and emperors so that in glory and triumph they could become the momentary masters of a fraction of a dot. Think of the endless cruelties visited by the inhabitants of one corner of this pixel on the scarcely indistinguishable inhabitants of some other corner. How frequent their misunderstandings, how eager they are to kill one another, how fervent their hatreds. Our posturings, our imagined self-importance, the delusion that we have some privileged position in the universe are challenged by this point of pale light. Our planet is a lonely speck in the great enveloping cosmic dark. In our obscurity, in all this vastness, there is no hint that help will come from elsewhere to save us from ourselves. The Earth is the only world known so far to harbor life. There is nowhere else, at least in the near future, to which our species could migrate. Visit? Yes. Settle? Not yet. Like it or not, for the moment, the Earth is where we make our stand. It has been said that astronomy is a humbling and character-building experience. There is perhaps no better demonstration of the folly of human conceits than this distant image of our tiny world. To me, It underscores our responsibility to deal more kindly with one another and to preserve and cherish the pale blue dot, the only home we've ever known. Thank you. Sorry I didn't bring apples for all of you. I just couldn't carry them. Yes?
With all the research with some of these organisms, are there some safety measures as far as being sure that we're not researching so enthusiastically that we're causing extinctions as well?
Excellent question. Those cone snail shells that you got passed out, some of those species are in danger. Probably not the ones in your bag, but some of them are very similar in appearance to ones that are. There's a vast curio trade in selling shells. Cone snails are particularly beautiful. So people in the Philippines and other parts of Southeast Asia collect them and can sell them and endanger some species. Medicine is not immune from being irresponsible and collecting species to find medicines and research models. And there are examples of that. But there is a strong ethic increasingly so in the literature, that it's important to identify natural compounds in organisms, in natural ecosystems, but it's very important once you identify that to immediately synthesize it so that you're not having to extract those chemicals from the natural source and therefore, as you say, quite rightly, endanger the organism that you're trying to protect. And Taxol is a great model of that. So this compound was found in the bark of this tree, but you would have to harvest the bark from 100 trees for one dose for treating ovarian cancer. So you'd wipe out all the Pacific yew trees and that would be it. So there was a very great rush to synthesize Taxol, but also to find the active parts of that molecule and then attach semi-synthetic chains to that. And what was great about that is they led to a whole new group of compounds that are even more effective than Taxol. So there's a real pressure to make sure that you don't endanger the organism. But of course, that's sometimes hard to please. So it's a very important question. Yes?
When you talk about the frogs and the antibiotic substances that they produce, you said that there was no bacteria weren't becoming resistant to those antibiotics, and I was wondering if we know that for sure, or if there's not a evolutionary arms race going on, and the antibiotics are changing, but we haven't been studying them long enough to know.
Yeah, I mean that's a really good question. Do you all know about this fungus called the chytrid fungus, which is wiping out amphibians all over the world? So there's a big debate going on. Is this an invasive fungus? That is it coming from a part of the world, actually thought to be Africa, where the amphibians have developed a resistance against this fungus and has been imported by Xenopus, you know this Xenopus laevis, which has been widely used for pregnancy tests and for other biological studies, and therefore spreading the fungus to other parts of the world where the amphibians have do not recognize the fungus, have no ability to attack it. So there's that school of thought. And then there's another school of thought that says that actually chytrid fungi are very ancient fungi and they're kind of all over the place. And you can go to museum specimens and find them in healthy frogs in North America and other parts of the world. And are they getting the infection because, like an HIV/AIDS patient gets a fungal infection that kills him or her, and those fungi, Pneumocystis carni, for example, they're yeast-like organisms, we have in our lungs all the time, and they don't affect us at all. But if we're stressed with HIV/AIDS or some other stressor, environmental stressor, then our immune system gets dysfunctional and we get sick. So is that why these amphibians are dying? Climate change, pesticides, ozone depletion, whatever. But when you look in the fossil record, there doesn't seem to have been an event where amphibians have been wiped out in this way historically, as far as is known. Amphibians are a little hard to study because they break down much more easily, and there are not too many amphibian fossils. But as far as can be told, that has not occurred. So this seems to be a unique event. But it's a very important question you have, and I'm not sure anybody fully knows the answer. In terms of the chytrid fungi, even that is a very complicated story. And you may be absolutely right. There may be examples where these antimicrobial compounds don't work and populations get wiped out and then others fill in, and we just don't know. I mean, we just haven't identified those species yet, for example. But my guess is that these strategies have worked for a very, very long time, and we need to understand them better. And that's true not just for amphibians, but for all organisms that live in a soup of microbes the way we do all the time, and don't seem to get sick, and have what are called very primitive immune systems, because they're not our adaptive immune systems. They may be very primitive, but they've been very effective for a very long time. So maybe they're not so primitive. Maybe there are secrets there that really we need to understand. Yes?
Do you know anything about the disappearance of the bees, the honeybee?
Oh my goodness. I say oh my goodness because I have bees, and I've been writing some about that. But there was an article on the front page of the New York Times a few weeks ago that some of you may have seen that basically said, bee colony collapse disorder solved. We know what's killing them. They're getting a viral infection and a fungal infection combined, and that's what's doing them in. But then the question is, why are they getting a viral infection and a fungal infection suddenly? Again, there's no history that there's been a kind of massive dying out of honeybees. People have had honeybees for a very long time, hundreds and hundreds of years. So this seems to be a unique event as far as is known. So there's one school of thought that says, again, these are invasive pathogens. People are bringing them in from Australia or Israel or some other place and then affecting. And of course, we have models for that. So that may be what's happening. The chestnut blight, for example. The American chestnut tree was one of the major trees in New England forests. They were all over the place, gorgeous trees. I have a very old house. All the main beams in that tree or in that house are chestnut. Hard but light wood, very strong. There are no chestnuts now in New England forest because the chestnut fungus, a blight, was brought into this country. I think it was in New York Botanical Garden, and then over decades spread everywhere. We're seeing the same with the hemlock woolly adelgid, this bug that kills hemlock trees. So we have a model of an invasive pathogen that comes in. So that may be what's happening with bees. My suspicion is that that's not the story. That may be part of the story. But my suspicion is that bees are under other stresses that are affecting immune systems. So it's this HIV/AIDS model, again, that they're being weakened. Why are they being weakened? Well, many reasons. One is that, particularly in places like California, big agricultural areas, huge business to pack up all your hives in your trucks and truck them to the almond farm or to the orange grove or to somebody's apple orchard or whatever. That's extremely stressful on the bees. So that's not a great idea. That may be one stressor. This summer was very hot and dry in New England. I don't know how many of you are from New England, but it was a very rough summer. My bees did not make enough honey this summer. Usually they make a lot of honey. And the reason is when it's hot and dry, flowers don't make as much nectar. So they don't have as much food to make honey. And that's a stressor. And also the temperature inside the hive gets very hot when it's 95 degrees for several days. And they don't have enough water available to fan the queen and evaporate water to cool the hive. So that must be a stressor, and there's good evidence that warming and climate change is a stressor. I think the big culprit is probably pesticides, because particularly a pesticide called, which the group is nicotinamides, and the particular pesticide is called imidacloprid. And it's a systemic pesticide, so when you treat a plant, the entire plant gets the pesticide. So it's not like spraying on the surface. Inside all the fluids in the plant have this pesticide in them. So it comes out in the pollen, so bees are exposed. It comes out at what are called guttation droplets at the edges of leaves, and they've measured the concentration of imidacloprid in these droplets, so bees drink water from those droplets, so they're being exposed that way. It's a known neurotoxin for bees, a lot of literature. It's banned in France, Italy, and Germany. It's made by Bayer Crop Science. They're probably going to sue me, but I'm okay with that. You can come after me, guys. And they've been trying to discredit this research for a very long time, lots of cases in court. It turns out, and I wrote a letter to the New York Times that they published about this, the lead author of the study on the front page of the New York Times, Jerry Bromenshank from University of Montana, who said bees are not dying from pesticides, they're dying from these infections. He has been getting money for his research from Bayer Crop Science. And he didn't reveal that to the journal he published his article in, and he didn't reveal that to the New York Times. So you got to smell a rat somewhere. Anyway, he's in deep trouble now because now that's all out.
I have one more question.
Yes,
regarding Lyme disease. If you do detect it early, is it pretty much curable?
Absolutely. Absolutely. One of the great things, if you can take doxycycline, it's a tetracycline derivative, there's very good evidence that in the first, I can't remember whether it's 36 hours or maybe even be 48 hours. It's somewhere between 24 and 48 hours after a tick embeds itself and is carrying the Lyme bacteria. If you take doxycycline within that period of time, it's only like one dose. I think it's 500 milligrams. Your chances of effectively treating the Lyme bacteria are over 95%. So that's why I do it. It's a very effective thing to do if you feel that there's a good chance you've been exposed to Lyme disease. In the early stages, it's essentially totally curable. It's when it becomes systemic after, months and even years that people then go on antibiotics for months and months at a time, and it's not clear sometimes if that's going to really work or not.
Yeah. So with ticks, I might have the same question. Say if you pull out a tick, do you just go with that, I think it's a 200 milligram dose, or would you go on it for a week?
I actually don't. There's good literature that one shot, one dose. Again, you go online and check it, but check initial exposure to Lyme doxycycline dose. I think it's like 500 milligrams once. There's very good literature.
5 capsules then, or?
I'm sorry?
That would be like 5 capsules?
No, I think they're either 250 or 500. I'm sorry, I'm not remembering. But no, you don't have to go on it for a week. If you have Lyme disease, then that's a different story. This is as a prophylactic.
Yeah, I'm always paranoid when I get sick.
Oh, I don't blame you. No, I am too. I have I have two very good friends, both big strapping guys who work outside all the time, unbelievably strong. One had Lyme meningitis, and the others had headaches and isn't quite right for 2 years now. And boy, they couldn't be healthier or stronger than these guys. So it's a serious business. You don't want to mess with it. Yeah.
I don't know how much you've heard of this, but I had heard something about, it was, I thought it was some kind of political, whatever, targeting at scientists who were trying to do either global warming studies or something, or it is existing, or all the things having to do with global warming, and trying to stop funding for the research for those people. Have you heard this?
Oh, man. I don't know if you want to hear what I'm going to say about this. But I mean, you've heard from my political jokes where I am. The New York Times finally on the front page reported that $500 million has been spent since 2009 by oil companies, coal companies, and utility companies to both block climate change legislation and discredit climate change science. So the two areas that have been focused on have been these emails from a climate institute, study institute in England, in East Anglia, where some climate change scientists were exchanging emails and said some pretty negative things about some climate change scientists, one of whom I know, who's at this university, whose name will not go mentioned, but has published really bogus stuff. And they said we should not allow this person to publish in the literature. That was jumped on as an example of trying to block science and trying to skew the results. So many investigations have shown that that's been bogus. So you have to wonder who's hacking into emails and publicizing this as a way of discrediting climate change science. The other was the fact that there was a mistake in the IPCC, the Intergovernmental Panel on Climate Change, that said that a glacier in the Himalayas was going to melt completely by 2035. It was supposed to be 2053, and it got transposed. So that was seized upon. How can you trust the Intergovernmental Panel on Climate Change, which has thousands of pages of data, thousands of scientists? Yeah, of course, there's a typo. As proof that this is all bogus and a myth, as many of the people who've just been elected to our Congress have said. But at the same time, try to ask yourself these questions. What's in it for a scientist to deliberately misrepresent the science? Scientists love to get their article on the front page of Science saying this article is completely bogus. It's very dangerous in the scientific world to deliberately misrepresent your science. What's the payoff? Oh, well, you may get another research grant, but what's the payoff for discrediting climate change science? We're talking about trillions of dollars at stake. If there are controls about greenhouse gas emissions and moves to alternative energy, who's going to be affected by that? Ask yourself that question. And what difference does it make what's in the IPCC about glaciers in the Himalayas melting? We know that glaciers are melting in South America. We know that Greenland is melting. We know that the Arctic ice sheet is melting. You can see that. You can measure it. You can look at it from a satellite. We know that temperatures are increasing. This year is probably going to be the warmest year on record since 1856 that's been recorded globally. So there's been an attempt to distract people from what's really happening. And I think we're going to see more of that in coming years. So be vigilant about that. Because the stakes are very high. Stakes are very high. If Greenland melts, it's melting now. If the temperature's warm, we can be inside our houses and we can adjust our temperatures in our houses, but organisms in the wild can't. They are adapted to specific temperature zones. We see what's happening with coral reefs. We see what's happening with polar bears. They depend on an environment that they have evolved in and adapted to for very long periods of time. What happens when we affect those ecosystems in basic ways that supply us with food, that affect infectious diseases, that affect our lives, those changes are huge. So be aware about that.
Thank you.
You're very welcome.